We're also targeting a wide range of grades of OA, so we're looking at KLs 2 through 4, and of course, we, for safety reasons, pregnancy is excluded, and therefore a test needs to be performed. So the design of this study is to first do a screening examination. We're going to do lab testing. We've been doing lab testing, X-rays, and the MRI, and it is a 1-to-1 to 1-to-1. And so the first arm is a bone marrow concentrate, or BMAC, versus corticosteroid injection. Second arm is to do an SVF injection versus a steroid injection. And the third one is allogeneic, and we opted for the human umbilical cord tissue-derived MSCs compared to steroid injections. So the umbilical cord MSC procedure, these are cells that are received from the Carolinas blood bank at Duke University. Each batch of cells or cell doses undergoes a battery of analyses prior to release, with a certificate of analysis accompanying each batch. We perform an on-site release testing. We do cell counting, cell viability, and we also do some tube inoculation, sterility tube inoculation tests. After all release testing results are acceptable or deemed acceptable, we do some injections. And so for the autologous cell harvest, we got a BMAC versus SVF. So the BMAC is about 60 cc's of bone marrow that's aspirated, and we inject about 7 cc's of BMAC under ultrasound guidance. And for the SVF, we get 35 to 40 cc's of lipoaspirates, which then yields these SVFs, and we inject about 5 cc's into the knee joint. The clinical outcomes that we're looking at are within 12 months, as I said previously. And so we have patient-reported outcomes, KOOS-BAS, and equality of life at 1, 3, 6 months, 9 months, and 12 months. And we are performing 2 MRIs in addition to the initial one upon diagnosis, 1 at 6 months and 1 at 12 months, so that we can do some cartilage mapping, hopefully. And so what makes this study a little bit intriguing to us, and the reason why I was extremely interested in it, is to be able to do cell characterization. And so once we collect the cells from these patients, what we're able to do is look at their characteristics. And so in collaboration with the MC3M from Georgia Tech, we're not only looking at viability, but we also want to look at phenotypic markers. These are surface markers. We can do that by flow, but we're also doing some single cell transcriptomics, not only to define the subpopulations that are present in these injected cells, but also to look at transcriptomic landscapes within the single cell at the single cell level. We're also planning to do some lipidomics and proteomics. So a molecular characterization of these cells is going to enable us to see and hopefully at the end of this trial be able to correlate between the product that we're putting and injecting into these patients and whatever these cells are and what they're producing, hopefully. So far, we have recruited 312 patients. We have 168 left in the different sites. There are 2 sites in Sanford up there. This is pretty much what we have about this particular trial, and hopefully I'll be able to tell you, hopefully we will be able to tell you more as soon as we start analyzing these data. So I will use a few minutes to talk to you also about some preclinical work that's done in my laboratory, and hopefully it will generate some interesting questions. And we've been interested in stem cells. I'm most importantly interested in induced pluripotent stem cells and what their chondrogenic capacity is. And so we were able to derive some mesenchymal progenitor cells from these iPSCs from skin fibroblasts. But what we were able to see also is that if you get these cells from different tissues, whether it's cord blood tissue or from articular cartilage, what you see is that they do not have the same chondrogenic capacity. But one thing that we didn't expect was that if you take cells from human diseased OA discards and you reprogram these cells and generate some induced pluripotent stem cells, what you're gonna end up having, these are just on the right side a demonstration that we have obtained pluripotency and completely reprogrammed the cells. Although you do see that the surface markers are pretty similar between normal articular cartilage and OA-derived iPSC MSCs, that the ability of these MSCs, once they have been exposed to this inflammatory mayhem within the knee joint to be chondrogenic is decreased. And so based on this, we started thinking, what is the reason for that? And you would assume that reprogramming would reset all the clocks, and obviously it is not. And there is a wide range of literature out there showing that there are epigenetic changes that occur during osteoarthritis. And one of the genes that we wanted to look at, which is cathepsin K. This is a manuscript that we published looking at cathepsin K knockout mice and changes in their cartilage and subchondral bone in OA. That cathepsin K is abnormally expressed by chondrocytes in the osteoarthritic tissue. And so what we decided to do was to look at least at the methylation status of the cathepsin K promoter between MSCs that were from normal articular cartilage and an OA cartilage. And what you can see here is clearly that for this particular gene, there's hypomethylation, which means that this gene is still being expressed at a higher level, even though we reprogrammed the cell. So this is putting out there the idea that there, in addition to an epigenetic memory of tissue of origin, there may be also an epigenetic memory of disease that we need to be mindful of, especially when you think about all the work that has been done using autologous cells that were expanded and re-injected for many decades in the past. And so what we assume to be normal cells or healthy cells because they were from a non-load bearing area, may be actually not as healthy as we think. Finally, I want to talk about cartilage regeneration, and so very briefly talk to you about this work that we have done using another type of stem cells. In this case, it's human embryonic stem cells. And what we wanted to do was to see whether the pluripotency of these cells would give us an MSC population with enough plasticity to be able to push towards a hyaline cartilage forming matrix type of cells. And so the idea that was put in here was, what if we pushed them just a little bit towards making enough type 2 collagen, and then we used Wnt5a, a non-canonical Wnt, which we had demonstrated in the past using genetic models and and other models to be an inhibitor of hypertrophy or terminal maturation. So when we derived MSCs from these human embryonic stem cells, again, you can see in comparison with the gold standard, the bone marrow derived MSCs, that there is a relatively, I would say, comparable expression of markers or surface markers of MSCs, and they definitely are going to exhibit multipotency and differentiate into the different lineages, while they no longer express markers of pluripotency. So what we did is we treated them with for a short period of time with BMP2, again to initiate the chondrogenic response, and then we hit them with the Wnt5 later on. And as you can see here from this slide, where we looked at gene expression by real-time PCR, you can see that if you hit with BMP2 for 5 days and do nothing for the remainder days up to 14 days, type 2 collagen and alkaline phosphatase are going to continue going up. But upon treatment with Wnt5A, you can actually diminish or inhibit the differentiation of these cells. And so we took these pellet cultures and we put them in a nude rat defect that we create in the condyle, as you can see here, and with some fibrin glue, we then suture. And to make a very long story short, what we saw was that we had a very nice articular-like cartilage structure that was formed in the treated pellets, although the integration of this can be improved, and we are currently exploring the possibility of enhancing this with different, I would say, rehab and exercise regimens using these cells and many others. So I think what we have learned is that induced pluripotent stem cells are promising allogeneic source for MSCs, at least for cartilage repair. Would be really interested in knowing once the safety issues are removed, whether we can use them for also for cartilage preservation. Their tissue of origin, of course, matters, but whether they're previously exposed to inflammatory mayhem can influence their use for cartilage preservation or restoration seems to be a question that's worth asking in view of what we're discovering. We do have the ability to control the behavior and lineage commitment of pluripotent stem cells for large cartilage resurfacing purposes using growth factors and or small molecules. And the battle continues against post-traumatic osteoarthritis. So with that, I want to thank, of course, our sources of funding, the preclinical work that's done in my laboratory is primarily funded by the VA and the NIH and some funds from the Emory School of Medicine, and too many people to actually list. For this clinical trial. We're very grateful to the Marcus Foundation for providing us sufficient funds to be able to do a trial that would be considered to save us from the valley of death. And of course, our collaborators, Sanford, Duke, Andrews, and the Marcus Center for Therapeutic Cell Characterization and Manufacturing. And with that, I thank you.
That's great. Thank you, Hicham. Well, I look forward to the outcome of your study. That'll be a robust study. So we have 15 minutes solid for some discussion and questions. I think one point that comes out loud and clear in all the talks and is the need to characterize what it is we're putting in our patients. Hicham showed the extensive molecular characterization that you're able to do. I'd like to ask George. George, for the average clinician who's using cell therapy, bone marrow aspirate, who wants to contribute, what should we do? Should we send a small sample to the laboratory? Are there tests that your average, your clinician can do today in September 2020 on a cell formulation? Should we bank on aliquot? What are the markers of purity or potency or biologic activity that we might measure today? Any thoughts?
Help if I unmuted myself, wouldn't it? No, it's an important question and a practical question for clinicians, and I'm one, and I think the four things that I've taken away from this is, one is that it's important, as Arnie said, and I've tried to communicate that we stop using the word stem cell in terms of patient education. That the truth is that if we're injecting any of the cells that we're currently using, even the culture expanded cells, frankly, the least common cell we're injecting is a stem cell, is a true stem cell. They're cells, and we can measure them, but we should get away from the word that has now become too wrapped with hype and misunderstanding and potentially false interpretations of promises. The second is that if we're going to do this, as many of us have pointed out here, and particularly in the last talk with Dr. Drissi, is that we really need data. And so if you're going to do this, and I don't discount the idea that these therapies could be offered to patients with appropriate education, but you really want to collect data about the patients before, the patients after, so you can show that they're improving and know that you're contributing to evidence that you're helping people, and then also know what you're injecting. And it's not a trivial process of measurement. You could take samples and send them to the lab and get a cell count and get a platelet count. But It's not a trivial process of measuring the number of progenitors that are there, or the exosomes that are there, or the proteins that are there. And so it's really a question of do you want to make that commitment? And if you do, I would not recommend doing it by yourself and trying to reinvent the wheel. You would want to do it in collaboration with someone who's already doing it, who's already thought through the process, already has things that there's a path that you can follow, because it's expensive and it's possible to waste a lot of time and energy and money in restarting something that's not necessarily easy. As Dr. Drissi will tell you, this took him a year or more to get all these processes in place to be able to do what he's currently doing. And for the average clinician, it's too much time in a busy clinic. You'd be better off telling the patient, you might be a candidate, go to somebody who's currently doing this.
Right, and it can be cost prohibitive. I mean, the tests that Hicham's doing are great, but it's not practical on a widespread basis. Certainly, we need to get to the point where we have some sentinel markers that we can measure. I think Dr. Felsen, is he able to ask a question? Had a question.
Yes, thanks, Scott. I apologize for not for joining late, and if my question and comment was already covered earlier, please excuse me. But so, as someone who's reviewed this literature a couple of times and given talks about it, I guess I wanted to ask, was there any discussion, or ought to be there any discussion about there doesn't seem to be a lot of persuasive data that this is actually efficacious from a randomized trial perspective, notwithstanding all of the terrific and impressive science data that Arnie and others were presenting, where it might be quite effective, and uncontrolled and poorly controlled trials that suggested it might be. And I was thrilled to see that there was a large-scale trial that's actually been undertaken. But I wonder, if we don't need more large-scale trials that are well randomized and well controlled to sort of figure out whether one particular version of this is actually efficacious. And then that allows us to explore mechanism a little bit better than continuing to do small-scale studies that are underpowered to really ask this question.
No doubt we need rigorous randomized trials. Dr. Drissi, you can make some comments. You're involved
I think Dr. Caplan wants to take a stab at the question, and then I will go.
So actually, I want to make the long-standing comment that I've been doing this for 30 years, and the criticism sometimes I get is, Arnold, you've been working on this for so long. Why don't we have it in the clinic already? And so first of all, this is a very young, very nascent field. And although I'm very cocky and I know everything there is to know about MSCs, we know nothing. And there's two variables which are hugely important, which have been waltzed around by everybody, which I'm going to put on the table, is MSCs are produced in everybody's laboratory by a different technique, and indeed the commercial companies have their own criteria for how they manufacture these cells. And the issue is, except for a couple of limited cases that I know about, the manufacturing of these very therapeutic cells is not optimized for the clinical use for which they're being used. So that's number one. Number two is, it's even worse, is you take MSCs from me, which of course are the best you could possibly get, and you give them to Scott Rodeo because he has a rotator cuff problem. And Rodeo doesn't like my cells. So his genome is not receptive to what my MSCs is throwing at this injury site. So the two variables are optimization of cells for clinical use, and second is the response profile of the patient. And those are the pieces that we basic scientists are now working on. And so, for example, company Mesoblast, I have nothing to do with Mesoblast, but company Mesoblast just had an FDA public meeting where they were grilled, I mean really grilled, about this manufacturing issue. And as a matter of fact, they had a logic. They, in 2009, they changed their manufacturing techniques to assay for TNF-alpha receptors on the cell surface. And they're putting those cells into graft versus host disease patients who have huge immunological problems. So they at least optimize the cell production for the immunomodulatory capabilities of those cells. And that was a first step. There's other steps that need to be taken that those of us in the laboratory are working on. But the key issues for the gross and general dissemination of cell-based therapies are optimization of the cell being delivered to the patient and some way of analyzing the patient's response profile. And those are very difficult, both of those are very difficult modalities to do, but we're working on them. But given all of that negativity, we still see some very profound positive clinical effects on small studies. And again, we need these large controlled studies. And I want to make one last point, which is our expectation in this business is that there's going to be a large number of people who don't respond because of these two variables. But 30% of the patients don't respond to already approved drugs. Right. So if Scott Rodeo has a headache and he takes Tylenol and it doesn't work for him, he's a non-responder for Tylenol. Tylenol works for headaches. And so in the cell-based therapy, because we're still very early in trying to optimize all of this, there may be a higher non-responder group. And so therefore, placebo-controlled trials, for example, for COVID, I'm against them. I'm ethically against any placebo-controlled trial. We have non-responders. That's the floor on which we can measure therapeutic response. And so it's cell-based therapy, I think, is going to require different logics than all of us were taught in medical school by big pharma. So I think again, cell-based therapy working its way in. We're still early. It's starting to get a really good scientific basis. And we need these animal studies such as Lisa does, and we need large, well-controlled clinical trials with measurable outcomes to measure efficacy. So again, nascent technology, we're right at the beginning. Forgive us our mistakes, but realize in the last 30 years, we've come a long way. And one of the long ways we come is we don't call these cells stem cells any longer.
Those are helpful comments. And that really helps us to set the research agenda. I think you made two important points. One, you bring up the concept of precision medicine, right? The right drug for the right patient at the right time, and the need. And that kind of flows into their point I would make that I think you raised nicely is, in addition to all of our focus on characterizing what we put in the patient, the cell formulation, we need to characterize the patient, characterize the recipient, whether it's a blood test, look at the joint fluid like Hicham's doing. How do we characterize that patient? Ultimately, we can match. I think you implied that one size does not fit all, of course. And so how do we match the right formulation to the right patient, to the right pathology? Perhaps, Hicham, you can make some comments there, and in particular, with respect to the release criteria that have been determined for your umbilical cord cells, because that gets to this characterization of these materials.
So to just add a little bit to Dr. Caplan's answer is these large trials are necessary, but they're also cost prohibitive. They're very expensive. And so one of the issues, the main issue is that there are so many variables that one can look at a single time. And so if you imagine hundreds of patients to be able to do neck-to-neck comparisons, that becomes a little bit of a problem because it costs tens of thousands of dollars per recruited patient. And so I think that one of our friends from Sanford Health made the comment to everyone that Sanford has been paying for SVF trial RCT on their own, and so being able to obtain philanthropic dollars that would enable us to at least know what are the next questions that we should ask, because there's so many questions, and we're scientists, we're just kids in candy store. We want to answer so many questions. To be able to decipher the priority of questions that we want to ask is requiring this a large level of funding, which is not very easily obtained from the agencies that fund clinical trials here in the U.S. And so this is something we need to always be mindful of. In terms of release criteria, I think, in my opinion, in looking at these medicinal cells, the question that we ought also to ask is whether their homogeneity matters, or to what extent heterogeneity in what we inject is going to participate in the healing process. And so I am not so sure that the cell manufacturing field is going to be able to advance as fast as it could be without us better understanding what we're injecting and what level of heterogeneity do we need for other types of cells to support these MSCs, or, as Dr. Caplan elegantly demonstrated, how these MSCs will influence other cells. And so that's pretty much where perhaps my mind is going, and I'd be intrigued to hear what Dr. Caplan and Dr. Muschler think about this.
Actually, I might pick up on that. I mean, Dr. Felson's question implies the need for randomized trials and the placebo effect. So, but in veterinary medicine, we may not have the placebo effect. So maybe Dr. Fortier can comment from the veterinary perspective on what we can learn from their studies where there is no placebo effect, I presume.
Yeah, thanks, Scott. No, my patients don't talk, but their owners certainly do. Yeah, we have very we have very objective measures for our outcome, whether it's we have like GPS level lameness locators, so it's not really asking the patient how they feel. It's more, it's much more objective than that. So we've had lots of not very good, similar to human trials, and some not very good, some small studies. A lot of them are industry funded, which I'm not against. I'm a consultant to Arthrex and have done lots of other industry funded. The problem for us in veterinary medicine, if we really want to do a large study, which we are at Cornell, I know one of the other misconceptions for veterinary medicine is that we can do whatever we want, that we're rogue, but we really have all the same FDA requirements that human medicine does if you follow them. It's just obviously if a human patient is injured, it's much more egregious than in some people's minds than harming a human. So there's a lot more regulation on the human side, but the rules are the same. So at Cornell, we have 2 INADs. The A is animal drug. So we have 2 of those, one for equine, another one for canine. So we are doing large randomized clinical trials, and it depends on what the clinician really wants to do. I do mostly bone marrow cultured, some adipose. Some people are using micronized adipose tissue. But it's all autologous. I think one of the other things for all of these, how are we going to pick the cell and characterize the patient? We also still need to be looking at some form of antigenicity, whether it's complement or some other type of test, cross-matching, something like that. Because even if the cells are immunomodulatories, we all know, and they're MHC class 2 negative to start, they don't stay that way. So one injection probably isn't going to matter, but we need to figure out how to understand if that is clinically important before we really move forward with multiple drug doses.
Well, to that question, maybe we can, I guess we have 10 more minutes. We can continue here, which would be helpful because we could probably go on for hours, but the injection schedule, critical. So you injected your cells in your tailless model at 4 days. Maybe you and Hicham can talk about to us, how do we choose? Do we do 1 injection, multiple injections? What time frame between injections? I understand it's probably arbitrary, but any thoughts on if someone's going down this line of designing a trial or just using this clinically? Should we do 1 injection, 2 injections, 3 injections?
Yeah, for me in the clinic, what I do if I have a horse with a tendinopathy, we'll do an ultrasound examine, longitudinal and cross-sectional, and then I'll have a quantitative amount of their lameness with what we call a lameness locator. So it's not just me saying, oh, you're better, or the horse, as the owner, saying that. So for me, when an animal comes in and I give them an injection, if they're not, and then I want to see them again 6 weeks later, 3 to 6 weeks later, depends on how soon they can get back here. It's not easy to get to Ithaca. But if they're not 50% better in every one of those parameters, clinically, on palpation, on lameness, and an ultrasound examination, if they're much better but not 50% better, I will give them a second injection. If they really didn't have a response, I think those are the non-responders. So I don't give them a second injection. Maybe they need something else. If they're more than 50% but not great, for those I'll give another injection. So that's how I frame it. And one thing that we didn't talk about at all here is rehab, and the importance of obviously weight control and rehab and all those things. And those are, you can fix the biology, but if you don't fix the mechanics, we're not going to get anywhere really.
And that's another black box, the effect of mechanical loading on the biology. How does mechanical load being rehab, joint motion, weight bearing, a whole nother topic portfolio, a whole nother day. But I think for the clinicians, it makes sense to, there's no one size fits all. And I think you need to have the ability to vary your injection protocol and schedule based on the patient's response. So I would evaluate your patient, whether you evaluate them in a week or 2 weeks or 6 weeks, and make a decision as to a repeat injection based on their response or lack of response. So, George, the question that does come up a lot is, there are limitations in the current regulatory environment in the United States where we have limitations on how much we can, manipulate these materials. what is your perspective on the potential for changes in the regulatory environment, and will that be helpful in our ability to manipulate and process cell therapy formulations? I
You're
think you're
muted,
muted, my
George.
friend.
I'm a great fan of our regulatory process, and that, you know, currently we have the ability to, you know, to do processing on blood products and deliver in a variety of ways as long as they're minimally manipulated. And then the FDA expanded that in the 361 category to allow tissue products, so bone and cartilage and fat and other tissues, if they're minimally manipulated and they're used for homologous use, meaning kind of what nature intended them for, what their current use is in nature. And so that is rational, and it provides physicians with a great deal of discretion. Once you start to head outside of that, there's every reason to do studies, and you know, studies can be designed in
Oh, I think we lost George.
as Hicham is doing, and as we are doing as well. We have a prospective trial that's industry funded, but that's randomized, a 2-to-1 randomization scheme where patients may get PRP or not, and we can follow them prospectively. But there's also a place, and this is where, you know, if there are clinicians who are really keenly interested in becoming involved, I think there's also a place for rigorous prospective registries where there are existing therapies, for example, the use of a centrifuge or the use of a particular matrix that's clinically available, where a protocol can be designed and one or more clinicians can agree to treat patients the same way. And in a prospective registry like that, you're exposing patients to very little risk other than the risks of failure, but not the risk of harm. And you can measure in a relatively small number of patients, 50 patients or more, whether or not you're achieving a positive outcome. And so that's the walk before you run strategy in terms of doing clinical trials. It'd be interesting to see what David Felsen feels about that as well. But Hicham, did you do a registry before you started doing your clinical trial work?
No, we did not. So the other thing I would like to add to this is that there is an opportunity for us as a community to be able to help the regulatory system better appreciate differences. For example, there is this random number that 70% viability is necessary, otherwise it's not a good product, we cannot inject it. So 68 is not good. Right. And so we're gonna know, perhaps by designing other trials that are geared towards rigor, how we can actually provide regulatory systems with enough ammunition to make clear and safe kind of regulation for these trials to perhaps flourish and have more of them. So this is something that I don't know what you guys think of.
I think a registry, a national registry for cell-based therapy, eventually has to come online, because the bad actors are not going to register their procedures or outcomes in that registry. And the registry, if a registry is open to all, professional and lay, you can go online and you can see how a particular clinic is doing in a particular protocol. And as Scott said, patients come all the time. They walk in the office and they say, "Oh, I read online the following thing. What's your opinion, doc?" And the answer is, it's on the registry. Here's the outcome percentage of that particular clinic. And as I can tell you, bone marrow transplantation has been around for 50 years. for the last 25 years, there is a uniform registry. There's not a bone marrow transplantation done without going on that registry. So you know what's been done, how many cells are put in, and what the outcomes are. And so we have to catch up with the hematologists and aggregate our information and somehow get somebody to pay for a national registry. And that's what's, again, resource management in our business is the rate limiting commodity. So we're always begging somebody for money. I've been begging people for money for a registry for a very long time. And and there are 4 or 5 now registries. Some of them are commercially focused. Some of them are more generally focused. But eventually, Sweden, for every biologic and for every implant material, Sweden has a registry. So if you want a DePuy hip versus a Zimmer hip, you go to the registry, you can get all the outcome data that you'll ever want to see on every single appliance put into people.
Yeah.
And it's socialized medicine. We're not as sophisticated as that, but eventually cell-based therapy is going to have to do exactly what George says. We're going to have a basis for how we characterize the cells, how many we put in, what's the introduction route, and what's the outcome parameters. And we have to keep the patient's identity not violated. And so I think eventually our profession, medicine, science, is going to have to demand centralized registries. I can tell you, NIH ain't paying for it. They don't have the money. Private sector ain't paying for it. They don't want to spend the money. Marcus Foundation ain't paying for it. Philanthropy is hard to come by, complicated enough. So eventually we as a group, as was said, are going to have to come together, demand of our various organizations a registry. And I think, Scott, AAOS is talking about this at the highest levels. And if AAOS stands behind a cell-based therapy registry and has the right parameters, all of us will get behind that.
Yes. Those are great points, and we should kind of wrap up with that. But there's no better individual to make those points than Dr. Caplan. I mean, truly, a lot of experts on this in this panel, but it's arguably you're the father of this field from your seminal publications really 30 years ago. So thank you. Well, hopefully we've accomplished the goal here of both identifying and pointing out the just tremendous exciting potential in this area, but really the need for further work. Hopefully we set some questions that'll help define the research agenda for all of us moving forward. And I'd love to thank our 4 speakers. These are really international experts in the area in this field, and you could assemble no better list of speakers. So thank you very much.